father of genetics, Gregor John Mendel, by organizing series of national webinars. I tribute to Gregor John Mendel on his birth bicentennial. We salute him as a paragon of science. On this third webinar of the series, I warmly welcome you again and thankful to the Honorable President, Honorable Guest of Honor, Honorable Speaker, dear participants, for sparing valuable time with us. Jai Maharashtra, Jai Bhim. Thank you. Thank you.
Thank you, ma'am. Then I request Honorable Dr. S.B. Kengar, sir, Principal, Yashwantrao Chavan. Dr. Kengar, sir, please unmute your mic. Kengar, sir, can you hear me?
Sir, पुढचं घ्या.
Okay.
Sir, बहुतेक नाहीत ना?
Okay. Then I request.
Continue the program.
Then I request to Principal Dr. Shankar Lavre, sir, for convener speech.
Good afternoon, everyone. We have organized national webinar series on the occasion of birth bicentenary of Gregor Mendel, the genius of genetics. And this is the third episode of this national webinar series. We have with us Professor Dr. Arvind Kumar Sinha, former Vice Chancellor, Netaji Subhash University, Jamshedpur, Jharkhand. He is the Secretary of Mendelian Society of India, that's International Journal Mendel. As well as we have with us Professor Dr. N.B. Gaikwad, Professor in Department of Botany, Shivaji University, Kolhapur. Dr. S.B. Kengar, Principal, Yashwantrao Chavan College, Science. More thing because Mendel has contributed. Mendel has contributed many things, and definitely we should know the things about genetics and all. So with this, we have with us Dr. Arvind Kumar Sinha, former Vice Chancellor, Netaji Subhash University, Jamshedpur, Jharkhand, as well as Dr. N.B. Gaikwad, Professor in Botany, Department of Botany, Shivaji University, Kolhapur. Dr. S.B. Kengar, Principal, Yashwantrao Chavan College of Science, Karad. Organizing Secretary, Dr. Sushma Kirtane. and all the participants. I welcome you all on behalf of both the colleges. Definitely, we are learning many more things from different webinars. We have organized already 2 episodes. Genetics Beyond the Mendel by Rupesh Bedarkar, then Pre and Post Mendelian Concepts by Dr. Pansetkar. And today we have a topic with us. It is a importance of mutations in crop improvement and crop protection. So welcome you all on behalf of participants, on behalf of our institutions and the organizers. Thank you.
Thank you, sir. Then I request to Miss Rutuja Bhosle ma'am to introduce today's guest, Professor Dr. Arvind Kumar Sinha, and speaker, Professor Dr. N.B. Gaikwad, sir.
I would like to introduce today's guest of honor, Professor Dr. Arvind Kumar Sinha, former Vice Chancellor Netaji Subhash University, Jamshedpur, and Secretary, Mendelian Society of India. Professor Arvind Kumar Sinha did his schooling from Zila School, Gaya, and graduated from Gaya College, Gaya in 1971. He passed the MSc examination in botany with cytogenetics and plant breeding as a special paper from Magad University, securing first class first position and was awarded gold medal in the year 1975. After that, he joined the University Department of Botany as a junior research fellow of UGC in November 1975, where he worked for nearly 2 years. And Professor Sinha joined the Magad University service in the capacity of lecturer in botany on 13th October 1977. During the service period, he was promoted to the post of reader in 1986 under merit promotion scheme and to the post of university professor in 1993. Professor Sinha served as the head of the Department of Botany at Gaya College, Gaya, and also founder head of the Department of Biotechnology till retirement. He was offered the post of Vice Chancellor of NSU, Jamshedpur, where he joined on 15th December 2018. However, due to personal reasons, he resigned from the post on 23rd December 2019. During 2013-14, he was made coordinator of NAC at Gaya College, Gaya, and during his tenure, Gaya College was accredited grade for A after this human resource development of Government of Bihar nominated Professor Sinha as a resource person of NAC, and he got the honor to address 18 principals of different colleges of Magad University and telling them about the procedure of NAC. Professor Sinha is credited with publishing 70 research papers in national and international journals. He has guided 27 students for their PhD degree. He has attended many seminars and conferences and presented research papers. He also chaired certain sessions. He organized many national and international seminars at Gaya College, Gaya, one of which is memorable, which was organized in February 2010. In this seminar, this excellency and visible man of India, Dr. APJ Abdul Kalam, was the keynote speaker, and Shri Nitish Kumar, Chief Minister of Bihar, was the chief guest. Professor Sinha is associated with many scientific societies such as Indian Science Congress Association, Indian Botanical Society, Society of Cytogenetics and Genetics, and Mendelian Society of India. He is also fellow of Indian Botanical Society and Mendelian Society of India. He is also secretary of Mendelian Society of India, which published an international journal, Mendel. He is secretary of Gaya College Alumni Association. Professor Sinha has been recognized by Rotary for social services, and he was best owed with Distinguished Secretary Award in 2005, Best President Award in 2009, and Outstanding Assistant Governor in 2010. And our next honorable speaker is Professor Dr. N.B. Gaikwad, Department of Botany, Shivaji University, Kolhapur, and Management Council member, Shivaji University, Kolhapur. His educational qualification is MSc, PhD, and NET. He has 22 years research experience and 20 years teaching experience at post-graduation. Professor Dr. Gaikwad is credited with publishing 55 research papers and 77 research papers and posters presented in national and international conferences. He has guided 8 students for their PhD degree and 1 student for their MPhil degree and guiding 2 students for PhD. He has completed 7 major and 2 minor projects, belongs to UGC, MOEF, DPDC, RGSTC, DAE, BRNS, DST, SERB. Professor Dr. N.B. Gaikwad published a new species, Crotalaria shrirangiana, family Fabaceae, a new rattle pod from the Western Ghat of India. He has visited International Atomic Energy Association, Vienna, for International Symposium on Plant Mutation Breeding and Biotechnology organized by FAO from 27th to 31st August 2018. Professor Dr. N.B. Gaikwad worked as a Secretary of Shivaji University Postgraduate Teachers Association from 2015 to 2017 and presently in charge Secretary of Shivaji University Postgraduate Teachers Association up to December 2020. He has worked as Senate member from PG Teacher constituency of Shivaji University Kolhapur from November 2017 to August 2022. He has worked as Management Council and Syndicate member on Shivaji University Kolhapur from February 2018 to August 2022. Professor Dr. N.B. Gaikwad sir worked as a member of Grievance Committee of Shivaji University Kolhapur from February 2018 to August 2022. And he has working as a sectional member in botanical section of Indian Science Congress from Western India region from 2022 to 2023.
Thank you, ma'am. I think Principal Kinga sir with us. So I request to Dr. S.B. Kinga sir, Principal Yashwantrao Chavan College of Science, Karad, for presidential address.
Hello, good afternoon to all. And first of all, I'm very sorry I was not present for the first and the second such program. And this is a nice activity. It should be percolated to all the students. And this is necessary to organize such kind of a national webinar series on a birth bicentenary of George Mendel, and everybody should know about what the work had been done by the Mendel, not only related with the plant development, plant mutation, but it is also most important in case of the animal mutation and animal development. So whatever the new varieties or new species get developed, these species developed and then what the application, what the uses, what are the importance, that all the things are goes to George Mendel because of all these things are basically given by the Mendel, and so that the every student, every faculty, even the non-life science faculty should know that what the Mendel work and what the Mendel different activities done, what about this thing. So about in case of this birth bicentenary of George Mendel, and this is the third, I think the third lecture or the third webinar, and this third webinar that will give an inspiration to all the researchers, all the teachers, and hope so in future such kind of the more activities should be organized in as the online mode, but it should be organized in offline mode so that the gathering of all the researchers, all the students will take place in the different colleges, different institutions, and direct interaction it can be takes place about the mutation, about the different things, and about the work done by the George Mendel. So, Department of Botany of Yeshwantrao Chavan College of Science, Karad, Principal Laure sir, and all the dignitaries, all the scientists, they have given the nice information, nice idea, nice things to the researchers, students, and the teachers regarding with the uh, birth bicentenary of George Mendel. Thank you for the organizing, and thank you for uh, all these things, all these such ideas which are to be organized, which are to be arranged by the different institutions, and in future, uh, such kind of national conferences should be organized in the different colleges. And I assure to all the dignitaries or the scientists today that are present, uh, I will be hosting the to organize such kind of activities in my college. Thank you for inviting me as a for the presidential address. Thank you.
Thank you, sir. Then I request to today's guest, Professor Dr. Arvind Kumar Sinha, sir, for his valuable guidance.
Thank you very much. Good afternoon to everyone. Shubh Dopahar. Tuma Sarvana Namaskar.
Good afternoon, sir.
Good afternoon. Chairman of the webinar, Dr. S.B. Keninger, Principal of Yeshwantrao College of Science, Karad. Convener, Dr. Shankar Laware, Principal, Arts, Commerce and Science College, Sonai. Organizing Secretary, Dr. Susma Kirtane, Assistant Professor, Department of Botany, Yeshwantrao College of Science. Speaker, Professor N.B. Gaikwad, Department of Botany, Shivaji University, Kolhapur. Academicians from different colleges, friends from the Mendelian Society of India, dear students, ladies and gentlemen. At the outset, I would like to congratulate the chairman, convener, and secretary for arranging lecture series on the occasion of birth bicentenary of the great scientist Gregor Johann Mendel, who is popularly known as father of genetics. I would like to tell the audience in brief about the great scientist Mendel. Mendel, a teacher and a monk from Brunn city, which was earlier in Austria and now in Czech Republic, created history in the field of genetics by making us aware about the inheritance of characters from one generation to other generation, that is heredity. Gregor Mendel, with vast knowledge of mathematics and natural science, worked on pea plant, that is Pisum sativum, from 1857 to 1864. It took several contrasting characters of pea plant, such as tall and dwarf, mid crosses among them, and obtained F1, F2, and other generations. On the basis of experimental findings, Mendel formulated 3 laws of inheritance, namely law of dominance, law of segregation, and law of independent assortment. He presented his experimental findings before the Brunn Society of Natural Science in 1865. This was also published in the Proceedings of the Society in 1866. But unfortunately, there was no response to his discovery until 1900. Many reasons are cited for this. At that time, many biologists like Darwin and others were concerned with the problems of evolution, and they were looking for gradual changes from one type to another, that is continuous variation. But Mendel's findings indicated an unacceptable constancy of hereditary factors. In addition, the types of characters Mendel used in his experiment were examples of discontinuous characters, that is tall or dwarf, smooth or wrinkled, and so on. Furthermore, Mendel's preoccupation with probability events and mathematical ratios was an unfamiliar approach to biology that did not excite much interest. Prevailing opinions maintained that biological phenomena were much too complex to yield to mathematical analysis. Further, Nageli, the famous botanist with whom Mendel corresponded, also failed to recognize Mendel's fundamental contributions and did not show any interest. Mendel, being disappointed, left the scientific arena and got himself engaged in the work of monastery. He died in the year 1984. Finally, in 1900, Mendel's work was discovered simultaneously by three scientists, namely Hugo de Vries, a Dutch biologist, Karl Correns, a German botanist, and Erich von Sermack, an Austrian botanist. There was an immediate appreciation and response to Mendel's finding, and they rapidly became known throughout the world. It was really unfortunate for Mendel that during his lifetime, his findings were not accepted by the scientific world. But at the same time, it will be proper to say that Mendel was really much ahead in the scientific temperament as compared to others. His discovery led to the beginning of our knowledge regarding heredity, and therefore Mendel has been aptly called father of genetics. I salute the great scientist. Today's topic of the webinar is importance of mutation in crop improvement and crop protection. This is an interesting topic, and in my opinion, close to Mendelism, as in Mendelism, we talk about natural variations, while in mutations we find created variations. We all know that mutation is a sudden change in gene which is heritable. For crop improvement, we create desirable mutation by physical mutagen or chemical mutagen or instructional mutagen treatments. Induced mutagenesis is one of the most efficient tools used for the identification of key regulatory genes and molecular mechanisms. It is a promising approach to develop new varieties with improved agronomic characteristics. When we talk about mutation breeding, we say that it is actually variation breeding, where seeds are exposed to chemicals, radiations in order to generate mutants with desirable traits to be bred with other cultivars. In this context, I remember great geneticist and plant breeder, Dr. M.S. Swaminathan, who developed an improved variety of wheat called Sarvati Sonara from the mutant produced for amber seed color in wheat. This is a specific example of the importance of mutation in crop improvement. Therefore, today's webinar is interesting and worth listening as it is related to the role of mutation, the importance of mutation in crop improvement and crop protection. I am very much confident that the viewers will be greatly benefited by the talk of the learned speaker, Professor Gaikwad of the Shivaji University, Kolhapur. With these words, I end my speech and give my best wishes to all of you. My sincere thanks to the organizers, especially Dr. Susma Kirtane. Once again, thank you all. Namaskar.
Thank you, sir. Then I request to today's speaker, Professor Dr. Professor Dr. N.B. Gaikwad, sir, for his session.
Yeah.
Is it visible?
Yes, sir. It is visible now.
1 minute, just. Am I audible?
Yes, sir.
Yeah. Good afternoon to all. Chairman of today's webinar series, Professor Kengar sir from YC College Karad. Convener, Principal Dr. Lavre sir from Sonai College. Organizing Secretary, Dr. Kirtane Madam. Honorable former Vice Chancellor, Dr. Arvind Kumar Sinha sir from Netaji Subhash University, Jharkhand, and presently working as Secretary of Mendelian Society of India. Fellow participants and student friends. At the outset, I am very much thankful to the organizers for providing this opportunity to share my views in the national seminar, which is organized on the bicentenary birth anniversary of Gregor Mendel. So in this particular webinar series, I will try to share my views or whatever my little experience on mutation breeding. So how that particular mutations are important in crop improvement and crop protection. From next 40 to 50 minutes, I will try to explain what is a particular mean by mutation, how that mutation can be induced, or what are the particular achievements, and how mutation can also use for crop protection. And lastly, I will share some of my experiences with the audience. Okay, there is some problem with slideshow. So what I will do, I will show the slides which are having some magnification. Okay. Now we know mutation. So the term mutation was coined by Hugo de Vries, where heritable change observed in the characteristics of that particular organism. So whenever that particular change is observed in the genetic constitution of an organism and that is heritable one, then we can call it as a mutation. And this particular mutation is because of action of agent, that is chemical or physical agent, or somewhere spontaneous mutations are also there, but it is not because of genetic segregation or genetic recombination. So that change should be observed or occurred because of action of chemical or agent or physical radiations, and that should be a heritable, that is from one generation to the other generation, then we can call it as a mutation. So this term was coined by Hugo de Vries in 1900. As far as types of mutations are concerned, so mutations are observed in nature under two headings, that is spontaneous mutation and induced mutation. So in that, spontaneous mutations are very rarely observed, whose rate is very, very low. And these mutations are observed naturally either by lightning or some environmental conditions are there. So because of which these are observed. Whereas induced mutations, where these are induced by the action of initially by radiations, that is ionizing or non-ionizing radiation, these were used, and after World War II, chemicals were used, and which are causing action on the genetic constitution of that particular individual, and from which we are observing different type of changes in that particular genetic constitution, and whose rate is higher than that of the spontaneous mutation. If you see induced mutations, so first it was reported by Müller in Drosophila, where he used X-rays, and how X-rays can induce changes in that particular Drosophila was the first evidence where it was reported, yes, we can do artificial changes in that particular genetic constitution by exposing to radiation. Later, Stadler has reported the effect of X-ray on in barley and maize. And then after the World War II, or during that period, Oryobach, she has used that particular chemicals, different chemicals, and reported, yes, chemicals can also be used as an alternative to radiations for induction of genetic change in that particular organism. Now, if you see that particular mutation breeding, so in that
Sir.
Have to see, and then we can say how that particular results in the form of change in the phenotype, change in the biochemical character, and so on. So all those will come under that particular. Am I audible? Hello?
Yes, sir.
Yeah,
Yes,
yeah.
sir. You are audible.
Yeah. So the such kind of particular changes are observed. Now, in that case, if you see that particular new heritable character is there in that crop, or how that particular crop is showing diversity, which is not with respect to a particular trait, which is not reported earlier in the germplasm stock, and so on. So that character one can create. So genetic variability can be induced by using that particular induced mutations. Now, as far as procedure of mutation breeding is concerned, so what we have to do, we have to select the particular plant material, then which mutagen or radiation we have to use, that we have to suggest, or that we have to finalize. Then actually we have to give the treatment of that particular plant material, and then we have to handle that material very precisely. So that is very important, how we are handling that material. And then we have to further select desired plant from the undesired one, and then we have to see its true to type behavior in successive generations. So these procedure, once you follow, then we can say that yes, whatever variability which we have induced or created through induced mutation program, and having heritability, that is continued from generation to generation, then only we can say it is a stable mutant. Now, selection of material, that is very important. So in that case, what we have to do, we have to induce mutation, no doubt, but for that purpose we have to select that particular variety or that particular plant type in which that particular variety is more popular, but becoming inferior for one or other trait. So keeping all those background, we want to just induce that particular, or we want to change that particular trait. So that first we have to decide, and then we have to start that particular breeding program. So if it is a seed or if it is a propagule, so as per that we have to select that particular material, or sometime even we can do that particular mutations in vitro also, so where we are using callus or that particular cell line, or even embryos and so on. So that also we can use for induction of mutation. Now in that case, whatever plant material we are going to use, that should be a pure variety, or it should be in the form of pure form and healthy seeds. If we are using a seed, then that seed should be pure and healthy one and uniform. So if at the selection level, if seeds are not uniform, not pure, or they are not healthy one, then that whatever procedure we are following further after induction of mutation program, so we may not say whatever changes which are occurred, these are because of either mutation, or these are because of whatever seed material, that variable material that we have used. So for this purpose, one should select pure seed, healthy and uniform seed should be there. Then once that particular plant material is selected, then which plant part one can use for that particular treatment. So seeds, pollen grains, in specific cases, pollen grains are irradiated, and then these are used for pollination. Then vegetative propagules are also used, then cones, bulbs, and complete plantlet, that is in gamma garden. So we are putting that particular plantlet in that gamma garden, and continuously that radiation is given, where that low doses of radiations are exposed for 8 days, 10 days, or 15 days, and then we are observing the effect. So normally seeds are commonly used, followed by vegetative propagules, and pollen grains are used for this particular purpose. Now, next one, choice of mutagen. So in that case, one has to decide the particular mutagen, which mutagen we have to select. 1 minute. Which mutagen one has to use for that particular induction program, or which radiation one has to use for that radiation program. So that we have to find it out either from literature or previous workers results. So that we have to decide, and then we have to select that particular material either in the form of seed or propagule or pollen grains and so on, so that we can start a particular program. Now these are the particular examples how that particular radiations or physical agents are used. and what are their particular sources, characteristics, and how they are doing that particular mutations. So initially gamma rays, X-rays were used. After that, gamma rays, neutron, beta particle, alpha, and so on. So among the breeders, so these gamma rays are more popular as far as induction of mutations are concerned, because they are having high penetration power, and these are showing effects which are expected after exposure to that particular radiations are concerned. Then chemical agents are there, or these are commonly called as mutagens. So most favorable chemical agents are alkylating agents are there, which are adding that particular methyl group to that particular DNA structure, either that nitrogen base or phosphodiester bond, or your sugar. So and during next replication cycle, they will get replaced by other proper base pair, and so on. So point mutations mostly observed, so that we have to understand while effect of chemical mutation on the DNA sequence is concerned. So among them, azide groups are there, then hydroxylamine, and so on. So these are commonly preferred by the scientific communities, along with this nitrous acid, acridine orange, base analogs. So these are causing different effects on that particular DNA structure, and which will bring about either point mutation or transition or transversion, and so on. So ultimately we observe the change in that particular structure. Now, once we find out which agent, that is physical radiation or chemical mutagen, has to be used, and so on, then one has to further decide how that particular treatment has to be given, or in other words, which concentration or which dose we have to use for induction of mutation program. So that is first decided, and for which we are calling it as a determination of LD50, lethal dose. So that concentration or that radiation dose which will kill around 50% or more than 50% individual from that particular breeding program. So in that case, if we are using seeds for treatment with, especially with the help of chemical mutagen, then we have to use that particular pre-soaked seeds, or if we have to use radiation, then we have to send that dry seeds to the that facility center. So in Maharashtra, nearby facility center is Mumbai, BRC Mumbai is there, or even in Aurangabad or in Nagpur, that facility is available. So where we have to send that material, and we have to request them to expose that our seed material to so and so doses, and so on. Then once that particular treatment is over, then we are observing the effects. So in that case, lethal, sublethal, vital, and subvital. So in that case, lethal effect, so which the concentration or dose which is killing more than 50% in individual of that particular treatment, so that is considered as appropriate for induction of variability is concerned. Now, if you see that particular with the help of this chart, so once we are selecting that particular mutagen, then we are giving that particular treatment to the material, and in that case, that treatment after completion of that particular treatment, we are putting these seeds in the soil, and we are completing the generation that is known as M1 generation. So in case of conventional breeding, we are saying that F1, F2, F3. So in case of mutation breeding, we are calling it as M1, M2, M3, and so on. So we have to give the treatment, and we sow the seeds in soil, and then we have to observe the different parameters like chlorophyll deficient sectors, pollen sterility, then chromosomal abnormality by fixing the root tips, and so on, so that we can confirm, yes, whether mutation has induced in that particular plant material, and so on. Then we have to harvest the seed individually, and in M2 generation, we have to sow those seeds on plant to row basis. And where actually we observe that particular plant which is showing variability compared with the control or mother plant. And then we have to observe the desired plant material in M2 population, and then again those plants are harvested separately. And then what we are doing, we are confirming whether that character which we have selected in M2 generation, whether it is having 2-2 type behavior in successive generation, that is M3, M4, and so on. So it will be called as a 2-2 type plant, or if expression remains stable for generations together, then we call it as a stable mutant. And then that particular stable mutant can be used further for seed multiplication, multi-location trials for 3-4 years we have to do that multi-location trials, and then we can release that plant material as a improved variety, mutant variety. Or if that mutants are stable, then these mutants are further evaluated directly, or this can be involved in that particular hybridization program as one of the breeding parent. This is about mutation program through seeds. Now, if we want to induce that mutation program in vitro conditions, so what we have to do first, we have to establish that particular in vitro culture in the form of callus or that particular microbial culture and so on. So what is our requirement? As per that, we have to start that particular in vitro culture prerequisite, and then we have to expose that particular material to radiation, or we have to give chemical mutagen treatment with appropriate concentration, and then we have to observe how that particular plants they are behaving differently compared with the mother stock, and then we have to select the variable plant from that one. Now, in that case, if you see further mutation types, so in that case, these are broadly divided into intragenic or point mutations, structural mutations. What are these? So where that particular mutation which has occurred because of chemical mutagen or radiation, so in that case, it will change the DNA sequence. So one base is replaced by other one. So that point mutation is commonly observed in case of chemical mutagens, whereas structural mutations and other things where inversion, translocation, duplication, or deletion. So these are observed in chromosomes. So mostly radiations are involved in case of large alterations, or somewhere chromosome number is also getting changed, that is aneuploidy or haploidy or polyploidy, and so on. So such kind of particular changes are observed because of that particular mutations. And sometimes it is also observed that always mutations are not observed in the nuclear material. It is also observed in the cytoplasmic organelle, that is chloroplast or mitochondrial genomes are also there, which are getting affected by that particular mutagens, and based upon that we are observing the changes. And as far as appearance is concerned, so these particular mutants are observed, those who are able to observe the character visually. So such kind of particular mutants are known as macro mutants, or these are governed by many few genes, and micro mutations. So where characters are not visually observed, for which we have to use statistical tool for analyzing that particular changes which are observed in that particular part. Now, as far as applications of mutation breedings are concerned, so where one can say that this mutation breeding can be useful for improved crop varieties, or to induce, say for example, in case of conventional plant breeding, we require male sterile line in order to avoid that particular consequences which are associated with the emasculation and so on. So if male sterile lines are there, then breeders are very happy to start that breeding program. So that male sterility can be induced through this particular mutation breeding, or we can create additional genetic variability which is not reported earlier in that particular germplasm stock of concerned crop. And moreover, one can again create that particular crop plant which is having more adaptability to diverse agroclimatic condition. Now we are observing the effect of climate change on crop production. So intermittent rain, or high temperature, or low temperature. So in that case, if our plants are not able to cope up with that particular adverse environmental situation, then it becomes difficult to feed the population of our country. So how we have to overcome that particular situation? So where we can have an option of creation of variability through induced mutation. So that is making adaptability, plants will become more and more adaptable to that particular changing climatic condition, that we have to also find out. So it is a particular just list how one can induce that particular plant types which are showing resistance to different biotic stresses. So these are the particular examples from India and abroad, so which are showing resistance to different diseases. So if you see that particular cereal, pulses, oilseed, then vegetables and so on, so they are showing resistance to fungal diseases, bacterial diseases, then viral diseases, as well as some weeds are also there. Phenogamic plant parasites are there. So to these particular aspects, so plants are showing resistance. Then if you see that particular abiotic stress, not only biotic stress, through mutation breeding, one can create abiotic stress tolerant plant or resistant plants where drought is the main problem in our country. Then again, excessive irrigation and fertigation result into the salinity. So to overcome that particular problem, we require salinity tolerant plant and so on. Then adverse temperature are there, and it is not all over the country, but to certain extent at the hilly area, cold temperature is there, or a high altitude. So plants, how plants will able to complete the life cycle. So all those particular aspects were considered as far as creation of variability is concerned. In case of rice, then lentil, maize, and so on. So on one hand, we are trying to improve the quality, as well as on other hand, we want to improve the quantity also with respect to this particular abiotic stress. Along with this, again, how that particular mutation can create variability with respect to maturity or grain yield or grain quality. If you see that particular castor or rice or soybean, so where 30 to 45 days early maturity was reported in certain nutrients of rice, then castor, so that famous variety is there. So 270 days of maturity was reported. So that plant matures 150 days earlier compared with the control, then soybean and so on. So what is the application of this early maturity? So our field will get ready for next year, next crop, next season crop, that is Rabi, Kharif and Rabi. So if availability of water is there, one can take second crop or even third crop. So if crop life cycle get shortened one, so farmers are again able to take next crop based upon availability of irrigation facility and so on. Then grain yield, every farmer is expecting higher yield in his or her field. So if we provide a plant type which is having higher potential, then definitely every farmer will try to use that particular plant material which is developed from the breeding program. Similarly, grain quality is also there. So we know famous example as Sinha sir told that the contribution of Professor M. Swaminathan, that is Sharbati Sonara and so on. So how that particular single plant type which is developed through this mutation breeding program, so red color to amber color seeds were developed in case of wheat, and then all the consumers, they were preferring that particular seed and so on. So how that particular grain quality is also improved, that is achievement of that particular mutation breeding program. Then if you see that particular somewhere these particular plant types which are improved, these are further used by breeders as one of the parent in that breeding programming from wheat, barley and so on. So that is again example. Now if you see that particular global impact of mutants worldwide, then just because of rice mutation, how that particular release of that particular variety, mutant variety has changed that particular cultivation practice, area under cultivation, and the benefit which we are observing. So how much that particular enhancement in yield with 16 or more than $1 billion were observed in case of braid wheat, durum wheat and so on. So just because of development of one improved line through mutation program, it has changed the scenario of agriculture pattern, or the what we can say, that particular benefit for the farmers. It has provided that. Then if you see that particular India, Pakistan, Japan, and so on. So how that particular cotton, which has changed the income, additional income, or provided additional income to the farmers. Then area under cultivation has increased like anything. As far as Indian scenario is concerned, till today, so this is the data from March 2022. So 345 mutant varieties are released in India from different crops, and maximum mutant varieties were released from flower, so dahlia, tuberose, chrysanthemum, and so on, bougainvillea. So this work is from NBRI Lucknow. And then next to this rice crop is there, so where maximum mutant varieties were released, and these total 345 varieties were released from around 50 plant species. if you see further, these are the particular details of that particular 345 variety which are released from different crop plants and so same. Now, just how that particular investment on mutation breeding program has given return in case of black gram. I would like to put this particular example. So this particular mutant variety was released from TAU, that is Trombeacola udid, that is black gram mutant variety. So this particular before release of this particular variety, area under cultivation was very less, around that average yield was 419 kg per hectare. But after 10 years, that is that release of variety, that particular enhancement in yield was reported by 212 kg per hectare. And because of this, how much annual income has increased? Annual production around 1294 metric ton additional income was reported. And if you see that particular additional income, so around 25 crore and so on. So that is just because of one variety, and for which how much was invested? 1 crore. So 1 crore amount was invested, but it has given much more than that. Now, if you see that particular mutant varieties released up to 2022, that is latest data of 22 March. So in that case, around 3,400 varieties from 220 plants released worldwide. In that case, Asia, as far as area is concerned, Asia stands at first position, followed by Europe and so on. As far as crops are concerned, cereals where maximum number of mutants were reported, followed by legume, oilseed, ornamentals, and so on. And as far as agents which are involved in that particular mutant variety or improved variety, so physical radiations are or has played a very important role as far as development of mutant varieties are concerned. So this is about that particular data. And as far as mutant release worldwide up till now, so this particular data is there. So rice is having highest number of mutant release worldwide. Now this is about how mutation can be used for improvement of crop plant. Now we'll see how mutations are also useful for crop rotation, and then I will share my some experiences what we are doing from induced mutation in some crops. So insect pest control. So on one side, pest your microbes are also there, that is pathogens are there. So on other side, insects are also causing a loss in yield, especially cereal, pulses, vegetable and fruit crops. So we have to spray pesticides, and we know pesticide residues there, and again whatever residue is there, again it is coming to back towards us, either through that end product or through water and so on. So instead of that, is there any method through which one can control the growth of insects so that there will be no role of application of pesticide and on other side ultimately there is a reduction in the population of insects? So it will be a win-win situation. So in that case, that sterile insect technology is used. So what is that particular technique? So where we are using that particular male flies from that particular insect group, and these are irradiated, and then these irradiated male flies are released in nature. So what will happen? Because of irradiation of that irradiation of male flies, so they will become sterile when they will get met with the female. So female will lay eggs, but all the eggs will not able to hatch and will produce next generation. So that this is the particular technology. So what we have to do? So we have to take that particular larvae from that male, and then these will be irradiated, and then these particular irradiated male flies, which are sterile one, these are released, and these released male flies will mate, and then that female will lay eggs, but these eggs will not show that particular birth, or offspring will not be there. So ultimately birth control will be there. So this is a wonderful technique as far as controlling the growth of insects are concerned, which are causing huge damage to all type of crops in not only in our country but over the globe. So in that case, major achievements are there. So in case of Chile, Mexico, Tanzania, so just by exposing that particular insect male flies for radiation and release. So they have increased that particular production, and over 30 member states, they are using this particular sterile insect technology for controlling the growth of that particular plant. Then along with this, again, one can protect our food which is getting damaged during the storage condition, either through storage pests or through fungal infection or bacterial infection during the storage condition. So it is again very important to take care of food grains at the storage condition, or at the storage places where, because of moisture content and so, or improper storage conditions, we observed that particular insects or your parasites or pathogens, they are showing their growth on that particular seeds. So in that case, what we have to do, so not only seed, even a vegetable or even your flesh and so on, so that can be preserved just by exposing to the radiations. So where we are using gamma radiation or electron beams or X-rays one, and in that case, what we have to do, so we have to irradiate that plant material in such a way that it should not get damaged quality wise, but whatever exposed material is there, that exposed material will not allow the growth of that particular pathogen or insect or larvae and so on, which is present in that particular freezer or storage condition. So for which, what we have to do, we have to use that particular radiations with very low dose. So because of that, using low dose, what we are observing, so if you see that particular onion, potato, garlic, so these are the tuberous plants. So sprout inhibition, if we have to prohibit that one, then this particular dose we have to use so that it will inhibit that particular sprout. Then disinfection is there, insect disinfection and so on, or extension of shelf life period and so on. So all those particular aspect one can cover just by exposing that particular material with proper radiation with very small dose. So by this way, we are protecting our food grains or even our fruits or tuberous plants and so on from the infection during the storage condition. So as far as this application is concerned, so more than 60 countries, they are using that particular this particular practice for protecting the food grains from insect pests during the storage condition. And as far as 5 lakh ton is food are annually irradiated, it is estimated, observe that, and for which mostly cobalt 60, that is your gamma radiation, is used, which is having that particular ability to penetrate, and it will try to control the growth of. Now, these are the particular aspect which I have explained how mutation can be useful for induced mutation or importance of mutation in crop improvement and crop rotation. Now, I will give some of the experiences or first-hand information from our lab. So, what we are trying to do in Shivaji University, Kolhapur, and so from last I think 6 years we are trying to do this work, and now we are having some something in our hand. I will share my experience. So this particular aspect, that is genetic improvement of rice landrace through induced mutation. So Kolhapur district is known for few rice land races which are aromatic one, which are adapted in this particular area. But yield wise, very poor yield was reported, though quality wise they are at par with the basmati rice, but yield wise very poor. So farmers are very reluctant, though it is having market price, good market fetches good market price, but still because of unavailability of improved variety, improved lines of this one, farmers are not in a position to use this particular land races which are having aromatic one for cultivation. So we have chosen 2 land races, that is Azaragansa and BRNS program. So we have done this particular work, and now I would like to share this one. So this particular Azaragansa, that land race is very very well known in Kolhapur district as far as its cultivation is concerned. And what are the prominent features? So it is a typical local land race, not grown elsewhere in India. It is aromatic in nature, so production is less but fetching good financial returns. It is having medium grain size and desirable grain quality, and elongation is also very good compared with the basmati. It is having good aroma. So because of aroma only, we are preferring such kind of aromatic rice. So it is having good aroma compared with the basmati rice. And this particular rice required, or land rice required, a specific agroclimatic condition which are prevalent in that particular area, because of which it is confined to that area only. So considering this particular aspects, we also find out what are the challenges. So long maturity period around 140, 150 to 160 days, then it requires high rainfall, then less production is there compared with the other high yielding varieties of which are commonly cultivated in India. Then plant height is very high, because of which lodging is a common problem. Then less number of tillers are reported, and if you provide fertilizer, if you want to increase the yield, then lodging is a common problem. So considering all these particular challenges, we have started that particular breeding program under that DABIS program with these objectives, that is development of high yielding, lodging resistant, dwarf and early maturity variety of Azragansa through induced mutation. Now, for which we have used 2 chemical mutation and gamma radiation. I will not explain because of time constraint. I will go through this. So in M1 generation, we have see how that particular parameter, so different parameters were used, so which indicates that how that particular pollen sterility is there. Then chlorophyll deficient sector, so which indicates that how mutation has induced. Then these are the particular aspects where we are observing that particular chromosomal abnormalities are reported, so which indicates that yes, the mutation has induced. And then at the end of M1 generation, these plantlets were harvested separately. And in M2 generation, then we have sown those plant or transplanted those plant after 21 days after germination in plant to row basis, in order to see what kind of performance is observed. So during this, again, we have noted the chlorophyll deficient mutants in M2 generation, that indicates that yes, that particular mutation has observed in that particular line. So these were the observations or indicators. And then on plant to row basis, so one line, one plant indicates that particular one mutant plant, and like this way. So these were transplanted, and then we have reported that particular different chlorophyll, different sectors, and okay. So these are the particular traits which we have considered while selecting the desirable plant in M2 generation. And if you see that particular so dwarf plants, so our mandate was high yielding, dwarf, early maturity, and lodging resistance. So we have reported different plant types from different treatment, that is EMS, sodium azide, and gamma rays, with different 3 different concentration and doses were used. And based upon that, we have noticed some plants which are showing reduction in plant height, though yield was less, but the reduction in plant height was reported. Here also reduction in plant height there. Then maturity period was reduced by almost 25 days. And if you see that one, so compared to control, that yield was enhanced by 2 to 3 fold. So that was our observation in M2 generation. So these are the particular plants. So we are supporting our data with the help of this particular figure. So this is the control plant, and how that particular plant is showing reduction in plant height. Similarly, so these are the dwarf plant obtained from different treatments. Then these are the particular dwarf plants observed from other treatments of EMS, 1% treatment. Then look at this particular, how that particular enhancement in yield is there. Just because of enhancement in tiller number, ultimately yield was increased. So compared with the control, 58 and 64 tillers were reported from EMS 1% treatment. Then in case of EMS 1.2% treatment, look at that particular reduction in plant height. In that case, one plant was sterile one, we could not get any seed, but other plants which were showing reduction in plant height. Along with this, some of the plants, they show that particular reduction in days to maturity period by almost 23 to 25 days. So these are the particular dwarf and early maturity plants are there, high yielding plants are there. In case of sodium azide treatment, so where this particular treatment has induced majority of the high yielding mutants, so where we are observing compared with the control number of tillers and number of effective tiller. If you see this one, so almost four-fold enhancement in number of tillers and number of effective tiller, which has resulted into 3 to 4-fold enhancement in yield is there. Along with this, some of the mutants which some of the putative mutant plants were observed, which are showing 23 days early maturity compared with the control. So these are the particular plants. Look at that particular number of tillers compared with the control. So these are the plants obtained from sodium azide treatment. Then from gamma rays treatment, we obtain dwarf plant as well as high yielding plants, and early maturity plants were also reported. So these are the particular plants which are showing highest yield. So around 5-fold enhancement in yield is there compared with the control. So these are the particular plant types which are showing enhancement in yield just because of enhancement in number of tillers per pint, then enhancement in panicle length. Because of this, we observed that seed yield per plant was increased. And why? So number of seeds per panicle and so on. So all these were increased, and ultimately which has contributed into enhancement in yield. So these are the particular plant types which are showing enhancement in yield with higher number of tillers and effective tillers from 150 guy rate treatment, 200 guy rate treatment. Then along with this, our another mandate was there. We want to find out lodging resistant plant. So during that particular M2 generations, other plants, they show complete lodging. Some of the plants, they remain erect at the time of harvesting. So these plants are considered as lodging resistant plants, and these lodging resistant plants, they also show at par yield with the control. Now all the plants were harvested separately in M2 generation, and these were used in M3 generation for confirming whether whatever character which we have selected in M2 generation, whether same character is exhibiting in M3 generation or not. So these were transplanted in field in M3 generation, and on plant to row basis after transplantation, these were again screened whether that character is retained or not. So in that case, some of the so in total 56 mutants were obtained from putative mutants were obtained from M2 generation, and these were transplanted in M3 generation. And in M3 generation, out of 56, we have reported around 25 stable mutant plants were observed, which are showing true to type behavior. Remaining mutant, they did not match the their performance with the selected plants or mother plant. Now these are the particular plant types in M3 generation. So how that M3 generation plants were behaved. So look at this one. So dwarf plants, so that plant which we have selected, which having very less yield. Now in M3 generation, we reported that it is showing reduction, 50% reduction in plant height. Along with this, enhancement in yield is there. So that is the good achievement. So look at this one. So how that particular entire line is showing that particular dwarfness, continuous line. There is no variation. So then we can call it as a particular mutant. But still we have to confirm its performance in successive M4, M5 generation, and then one can say, yes, it is a stable line. So these are the particular plant types which are reported in M3 generation. Yeah, these are the another dwarf lines at this particular, at the time of maturity, we have taken the snap. So how that particular plants, they are showing continuously stable expression in that particular plant to row basis of that mutant line. Then in case of EMS, how that particular early maturity plants were reported, and along with early maturity, one of the early maturity mutant is showing enhancement in yield in M3 generation. So these are the particular plant types which are showing that early maturity compared. So this particular line, so nearby the control line is there. So how that early maturity line is showing superiority over the control. Then in case of high yielding, so high yielding, in high yielding, it is observed that whatever mutant plant which we have selected from EMS, gamma rays, and sodium azide treatment, so all the particular plants, they show 2-2 type behavior. No doubt that much enhancement in yield is not reported compared with the M2 generation. So 3 to 4-fold enhancement in yield is reported compared with the control. And why? Because of enhancement in number of tillers, number of effective tillers, then number of seeds per panicle length has increased, and along with this, again, days to maturity in some of the mutant plant has reduced one. So this is achievement. Then lodging resistance. in case of lodging resistance, it is observed that this particular plant remains stable. Along with this, again, they have shown enhancement in yield by 1.3 to 1.4 fold compared with the control. If you see that particular figure, so look at this one. So this particular plant, these are showing complete lodging resistance, whereas neighboring field. So this particular plant height is around 5 feet. So lodging resistant plant, they remain straight at the time of harvesting, whereas remaining plants which are showing complete lodging one. So that is about Azaragansa. So we have completed M4, M5 generation, M6 generation in 2022, and this year we have started our multi-location trial program along with the some state university, and we expect that in coming few years we will able to give something to the farmer from our mutation breeding program. Yet it is very difficult to say that yes, definitely whether we are able or we will able to give improved plant type for farmers, because for that purpose we have to go for multi-location trial and so on. So that this is from Azaragansa. Now second variety, that is Kala Jirga. So this particular variety or landrace is again cultivated in Kolhapur district. So through DST ACRB program, so this particular improvement program was launched, and what were the challenges? So same prominent features of Azaragansa, it is again around the commonly cultivated in Kolhapur district, especially Azra, Chandigarh, and that particular area. It is aromatic in nature, good financial return, but yield is very less, and it is having medium grain size and desirable grain quality. Again, as far as aroma content is concerned, it is at par with the your basmati rice, and again it requires these particular condition for completion of life cycle. Now, if you see the particular challenges of Kala Jirga, it is observed that maturity period is very high. Then it requires continuous rainfall up to the November. That is from June to November. It requires that rainfall, or at least water should flow continuously from that particular field. Then and then only that aroma will develop and it will retain. Its yield is very low. Then plant height is a common problem, so because of which lodging is there, less number of tillers are reported, and negative response to the inorganic fertilizers. So lodging is a common problem. So considering that, we have again started that particular mutation breeding program in that Kala Jirga, where we have used two chemical mutagen, that is EMS, sodium azide, and gamma rays with 3 different concentration and doses, and we have completed M1 generation. And in M1 generation, these are the particular field views. These are the particular chromosomal abnormalities which are reported, which indicates that yes, mutation has induced. Then these are the chlorophyll deficient sectors, which are the indicators of induction of mutation program. Then in M2 generation, we have transplanted those separately harvested M1 plant into field, and after 21 days, these were transplanted in the field, and on plant to row basis, once we transplanted, then these were screened for different aspects. So considering the different parameters, so such kind of puddling and other thing we have to carry out, and then we have to screen that particular plant. So in that case, these were the parameters which were used for identification of desirable plant types. And in M2 generation, we have reported the different plant types which are showing early maturity, then high yielding, and so on. So in this case, if you see that particular yield wise, so 1.5 to 2-fold, or some 2 to 2.5 fold enhancement in yield is there in some of the high yielding plants. Why? Because that number of tillers are increased compared with the particular control. These are the particular figures which are showing that particular how that particular plant yield or that particular height has changed. Then these are the particular observations from gamma radiations. So where we are observing enhancement in yield is in one plant where almost 2-fold enhancement in yield is there compared with the control. So these are the particular photographs which shows that particular how the dwarf plants, then high-yielding and so on. So these are produced there. Then in case of sodium azide treatment, again we observed that particular highest that particular plant yield was reported through 0.002% treatment of sodium azide. So this particular enhancement in yield and reduction in plant height was reported in that particular plant. In case of gamma rays, then these plants were harvested individually, and in M3 generation on first these seeds were sown on plant to rovices, and then these were transplanted in the field, and then these were again screened whether that particular 2,2 type behavior is expressed in M3 generation or not. So in that case, we have selected around 30 mutant plants, putative mutants in M2 generation from Kalajirga, and from that we have further screened the stable performance of these particular 30 mutant plants in M3 generation. So in that one of the dwarf plant was observed. So this particular dwarf plant, look at that particular height. So it is around 60% reduction in plant height was reported, and along with this particular plant is showing enhancement in yield. Around 1.5-fold enhancement in yield is there. So look at this particular plant, continuously it is showing stable performance. So that indicates that yes, plant is stable one in M3 generation. Then in case of your EMS treatment and sodium azide treatment for early maturity, it is observed that plant is showing around 8 to 10 days early maturity. Along with this, again, it is showing enhancement in yield just because of enhancement in number of tillers, number of efficient tillers, then that panicle length is also increased and so on. So these are the particular evidences which are supporting that particular parameter. So look at this particular early maturity. So how plants they are showing early maturity compared with the control. So control plants they still. So we have taken the comparative photograph for just one line of control and 2 lines of mutant. So one can find out how that particular mutants they are showing early maturity compared with the control. Then performance of high yielding plant. So in this case, high yielding plants which are showing somewhere reduction in plant height is also there, and along with this around 1.2 to 1.5-fold enhancement in yield is there compared with the control. So just because of enhancement in number of tillers and number of efficient tillers, it was possible. So these are the particular photographs which shows that particular high yielding particular lines which are observed in the field. And entire work, so this entire work was carried out at one location, that is government seed farm of Azra. Last 6 years we are performing our experiment continuously in same place, so that no one can say that yes, you have taken experiment or we have performed experiment at different location in different season. So same season, same location, and we are observing the stable performance of these mutants at this one location. So from this, one can conclude how that particular genetic variability can be induced through induced mutations. Yes, we have tried to create that variability. Then selection of mutagen is very important. So which mutagen with concentration or dose radiation, that is very important to start a particular mutation breeding program to induce genetic variability. And in our case, it is observed that moderate concentration or doses of mutagen or radiations are showing maximum desirable mutants in our present study, and differential response is also observed. So same mutagen when it is used, so these two particular cultivars, they are not showing similar performance. So differential response was observed in this case. So this is our particular observations. So I'm very much thankful to the funding agency, that is DRNS, then SCRB, DST, and even UGC also. One of the project was also there on soybean mutation breeding, which is this work I have not represented here, but I would like to acknowledge funding agency for granting some amount of fund to execute this particular work. And this is my strength. So these are my students who has actually worked in the field for last 6 years. They were there. So 6 years they used to stay at Azra location, and then remaining 6 months of a year they used to stay at Kharif season at Azra, and then remaining period they used to stay in the department university campus for further laboratory work. So this is Shital Desai, then Akesh Yadav, who has submitted their thesis recently, then Ranjit Londe, and this guy was earlier there, Akesh. Now he left and he joined somewhere else. So, but initially he was there with me. So just because of student strength, I could do this particular work. And now we have completed M6 generation of both the land races of Azaragansar and Kajirga, and with the collaboration of one state university, state agriculture university, we are doing that particular multi-location trial, and we hope in coming years will give something to the farmer as far as improved lines from Azaragansar and Kajirga. So with this, I will thank the organizer for giving this opportunity to share my views on the occasion of bicentenary birth anniversary national webinar organized by YC College, Karad. Thank you very much.
Thank you, sir. Thank you for your great session. Then I request to our technical team to share feedback link in the chat box, and also request to all the participants to fill the feedback form for certificates. And I invite to Miss Sukanya Yadav, MSc student of Department of Botany, Yashwantrao Chavan College of Science, Karad, for vote of thanks.
Respected President, Principal Dr. Kingar sir, Honorable Guest, former Vice Chancellor, Netaji Subhash University, Jamshedpur, Professor Dr. Arvind Kumar Sinha sir, respected Honorable Speaker, Professor of Botany, Shivaji University, Kolhapur, Dr. N.B. Gaikwad sir, Convener Dr. Lavre sir, Organizing Secretary Dr. Kirtana ma'am, all participants. I feel honored and privileged to get the opportunity to propose a vote of thanks on this special occasion. I convey deep regards and heartily thanks to our Honorable Speaker, Professor Dr. N.B. Gaikwad sir, for his excellent speech. He shared deep knowledge and thoughts with us, which are very much helpful to improve our knowledge about importance of mutations in crops improvement and crop protection. Today we got a very beautiful treasure in the world of knowledge because of you. Sir, I again very much thankful to you for this valuable speech. I thankful to Honorable Professor Dr. Arvind Kumar Sinha sir, Principal Dr. S.B. Kingar sir, Principal Dr. S.R. Lavre sir, Dr. Kirtana ma'am, each and every component of both the college for their efforts to make this event successful. I'm very much thankful to Professor Kiran Arangale sir, Ms. Rutuja ma'am, and all the respected valuable participants. If time means each and every second is money, then today you have spent million for us. Thanks to all of you for making this event successful. Thank you. Jai Hind.
Thank you, sir. Sinha sir,
Thank
thank
you.
you. Gaikwad sir, thank you.
Thank you, madam.
Thank you, Gaikwad sir. Thank you, Sinha sir, and all the participants for participating in this webinar. And I'll request Susma ma'am and Kiran sir to put the link in chat box. When you complete the entire feedback form, you'll receive the certificate. Thank you.
Thank you, sir. Thank you. Yeah.
Okay. Yes. So maybe within a minute, we'll end the meeting. Before that, just check the link in chat box. If anybody is having any question, you can just put the question in chat box. Definitely we'll try to answer it. Thank you.